Welcome back to Physiology Diaries, and today we're going to talk about yet another aspect of our physiology, and this specifically is a nephron. A nephron is the functional unit of our kidneys. Our kidneys are those two bean-shaped structures that are located retroperitoneal in our abdomen, and they're important for many functions, amongst which we have filtration of blood and also excretion of waste products of our normal metabolism, but also wastes from drugs and any other toxic substances that are ingested in the body. So this here is an illustration of a nephron, and there are two types of nephrons. We have cortical nephrons and we have juxta-medullary nephrons. So what is illustrated here is a juxta-medullary nephron. If you look at the structure of the kidney, it has an outer zone, what we call the cortex, and then an inner zone, what we call the medulla. So cortical nephrons are located entirely within the cortex of the kidney, while juxta-medullary nephrons actually project into the medulla. So this line that we see here in blue is the cortico-medullary junction. So whatever is above this line is the renal cortex, and whatever is below is the renal medulla. So let's start by appreciating the microanatomy of this juxta-medullary nephron. So here we have what we call the afferent arteriole. The afferent arteriole is a branch of the interlobular artery, and an afferent arteriole feeds into each nephron. And then this here is the efferent arteriole. The afferent arteriole brings in blood, while the efferent arteriole takes or drains away. Now, the afferent arteriole forms a complex capillary network, what we call the glomerulus. And after materials are filtered through this complex capillary network, they go into this space, which is called the Bowman space, that is shaded in black. And then from there, these materials end up in the Bowman's capsule, and then into the nephron tubule. So, in summary, the nephron has what we call the renal corpuscle. The renal corpuscle is a combination of the glomerulus and then the Bowman's capsule. And then we have the tubule, or the tube. And the nephron tubule is divided into a number of parts. So we have the proximal tubule, which is divided into the proximal convoluted tubule. Convoluted means it is folded or it is serpentine. And then we have the straight part of the proximal tubule, what we call the pars recta. So the juxta-medullary nephron will dip, or it will dive into the medulla. And so here we have a thick descending segment, which really has the same physiological properties as the rest of the proximal tubule. And then we have the thin descending segment, then we have a thin ascending segment, and then a thick ascending segment, which passes back into the renal cortex from the renal medulla. And then there we have the distal tubule. And the distal tubule has an early distal tubule right there, and then a late distal tubule, or the distal convoluted tubule here. And then from that we have the cortical collecting tubule, and the collecting tubule pours into or drains into a cortical collecting duct, this here, and the cortical collecting duct continues as the medullary collecting duct. So in a nutshell, these are the parts of this nephron. But also to appreciate, from the efferent arteriole, we have yet another capillary system that forms, or a vascular system, and this is called the vasa recta. This that you see in red running along the loop of Henle. The loop of Henle is this U-shaped loop that you see there. So the kidney has two capillary systems. It has the glomerulus, and it also has what we call the peritubular capillaries. But in the juxta-medullary nephron, the peritubular capillaries are modified into what we call the vasa recta, which is important physiologically, as we're going to see. So the other thing I want to talk about is something that is important in the normal physiological control, and that's what we call the macula densa, or a part of what we call the juxtaglomerular apparatus. So if we look at the early distal tubule here, there is a close connection between this early distal tubule and the afferent arteriole. They lie in close proximity, and that's important because it forms a certain set of structures which constitutes the juxtaglomerular apparatus. So here, if we look at this early distal tubule, within its wall we have specialized epithelial cells that form what we call the macula densa. This that I showed in shaded black there. And these macula densa are osmoreceptors, meaning they are cells that can scan or that can test the osmolarity or the concentration of fluid flowing within the tubule. And then from the macula densa, this cone-shaped group of cells is what we call the extraglomerular mesangial cells, also called the RAAS cells. And then lastly, within the wall of the afferent arteriole, we have specialized smooth muscle cells, which are called the juxtaglomerular cells or granular cells, just before the afferent arteriole enters the corpuscle to form the glomerulus. So these three structures, the macula densa, which are epithelial cells within the early distal tubule, then this cone-shaped mass of cells called extraglomerular mesangial cells or RAAS cells, and then the juxtaglomerular cells or granular cells. They're called granular because they have granules that contain renin. So these are important in what we call the RAAS, or the Renin-Angiotensin-Aldosterone system. In other words, any glomerular filtrate or fluid that reaches this point is supposed to have a particular concentration. And so these macula densa cells are constantly monitoring the concentration or the osmolarity of the fluid that reaches them. So if you have a scenario where an individual loses so much blood, for example, and becomes hypotensive, and we have a reduction in the renal blood flow, which of course reduces the hydrostatic pressure within the glomerulus, then we're going to have a decrease in what we call the GFR, or the glomerular filtration rate. And so that means that the tubular load, or the amount of sodium chloride that goes into the tubule, is reduced. And so the amount of sodium chloride that goes through the tubule to reach the early distal tubule to reach the macula densa is also reduced. So that reduction in the concentration of sodium chloride is going to be detected by the macula densa cells. And these macula densa cells, in response, are going to act via a paracrine signal to communicate or send a message to the juxtaglomerular cells or the granular cells, which granular cells now release the substance renin into the blood circulation. And then the renin cleaves or catalyzes the cleavage of a zymogen, or an inactive protein which is already present in the blood, which is called angiotensinogen. So from angiotensinogen we form angiotensin 1. Now angiotensin 1 is converted into angiotensin 2 by angiotensin converting enzyme ACE. And then angiotensin 2 has mostly three effects. One, it stimulates the adrenal cortex and specifically the zona glomerulosa to start the synthesis of aldosterone, but also angiotensin 2 exerts a constrictive effect on the efferent arteriole, and then the same angiotensin 2 also works to increase reabsorption of sodium along different parts of this tubule. And then the aldosterone also has other effects, among others increasing the reabsorption of sodium in the gastrointestinal tract, but also in the kidneys. So that is how the renin-angiotensin-aldosterone system works, and the resultant effect is going to be to retain more sodium, hence retain more water, and therefore increase our stroke volume and push our blood pressure back to the normal. So that is the RAAS system, and this is the functional unit of the kidney called the nephron. The other thing that I will talk about is something called the countercurrent system, and also about the medullary collecting duct and its important function as far as concentration of urine is concerned. So for now, I would like us to appreciate the nephron as a functional unit of the kidney. Thank you.